So hello everyone, welcome to my uh, video lecture on the topic in next generation sequencing analysis or NGS sequencing analysis. So in this video we will learn about a small theoretical aspect of how a next generation sequencing is done, and especially in case of Illumina sequencing, so you can get a basic idea of how this forward reads as well as reverse reads are generated in this case. So I will just show you a presentation. Okay. Okay, as you can, as you all know, basically you know that earlier times sequencing was basically done with the help of Sanger sequencing and all, and next generation sequencing is basically done in the process of Illumina, and nowadays like nanopore sequencing to completely read a whole genome is also being done. However, basically in this particular sequencing video, and we will be discussing about how sequencing platforms like Illumina sequencing and everything works. Basically, in nowadays also, most of the time Illumina sequencing is one of the most commonly used sequencing platforms, especially in case of research work and all. So, if you have anything like a genomic DNA, you can see that if you have a like organism like E. coli, basically you have a genomic DNA that is present within it, right? So basically, in order to know a whole sequence of the genomic DNA, then you have to extract the genomic DNA first, and then you have to fragment the DNA and do a next generation sequencing analysis or Illumina sequencing in this case. So how the whole process works in between, because it's a completely different process if you look into much depth of how a NGS sequencing analysis works. So basically what you do is that from the organism that you have to do a sequencing of the whole genome, you have to isolate the genomic DNA, because that's the prerequisite that we require in this case, right? And then what we does is that we have to fragment this particular DNA into shorter insert or small fragments of that case. So you can see that from this whole length of genomic DNA, it is getting shortened down into smaller fragments. So, and afterwards what happens is that to each end of the genomic DNA, right, of the fragmented DNA, you are adding a particular sequence is called as adapter. So adapter is particularly a short primer like sequence. You know that primer has is of short length, right? So it is similar to that. It is of short sequence, but it has a tendency to bind to the ends of the 5' as well as 3' ends of the fragmented DNA. As you can see there, this purple, I hope you can see this purple sequence is the adapter sequence, and it is bound to both the 5' as well as 3' end of the fragmented DNA by a process called as a ligation. To each of the DNA fragments, actually it is bound. So basically what you know is that for the fragmented DNA you don't know the sequence, but for the adapter you know the sequence basically at the both ends. So what happens is that then you after the adapter ligation and all, then you insert this fragmented DNA containing the adapter attached to its both ends to what is called as a flow cell. So this is a flow cell. Basically it is involved in Illumina sequencing platform and all. The sequencing machine is actually provided with flow cell. So the flow cell actually consists of some adapter sequence that are complementary to this adapter sequence. So in the flow cell, certain small adapter sequences are present there. You can see that this is a small portion within the flow cell. In that, the adapter sequences are present there, and it is conjugated or covalently bound to the flow cell through chemical interactions and all. And these adapter sequences have sequences that are complementary to the adapter sequence that have been attached to fragmented DNA. So that's why you can see that when it is the fragmented DNA with the adapter sequence is attached to the flow cell, the adapter sequence actually complement with each other and they bind to each other. And this process is called as annealing. It is similar to what a PCR reaction is having. There are three different steps, right? Denaturation, annealing, and extension. So in this case, when it is added to the flow cell, it undergoes a process called as annealing, so that the two complementary adapters bind to each other, and then it undergoes an extension reaction. So in the extension reaction, you can see that bases are added to the specific insert or the fragmented DNA to generate what is called as a read fragment. This is called as a read fragment whenever a particular a particular base are added, and from the insert that is bound to the adapter, whenever a new fragment is formed from the fragment that is attached to the adapter sequence, it is called as a read fragment. So this whole length is called as a read fragment there. So similarly, there are also other fragmented DNA with the adapter attached to it, they also bound to the adapters that are present in the flow cell and generate other different reads also. So this is how a sequencing works. Basically, from the fragmented DNA you are attaching an adapter, and this particular hybrid consisting of both the fragmented DNA as well as the adapter is added to the flow cell, and because of the complementary adapters that are present within the flow cell, they bind to each other by a process called as annealing. Then extension takes place to form what is called as a read. So afterwards denaturation happens, the old fragmented DNA is getting released from this particular adapter sequence, right, and it is washed off basically. So afterwards, when the if you look into a flow cell, it not only consists of adapter but also the polymerized also the newly generated read fragments that is attached to it, right. And what happens is that sometimes what happens is that in some cases the adapter sequence and both ends will be the same. In the fragmented DNA, in this case, it is both same, but in some cases one adapter sequence will be adapter 1 will be different from the adapter 2, so the sequence will be different. In that cases also the flow cells will be different. In this case there is only one adapter present in the flow cell. In that case another different adapter sequence or adapter 2 other than adapter 1 will be present here. Both will have a different sequence in that case. So anyways, that's a different case. In most cases both two different types of adapters are used within the flow cell. So in this case what happens is that once the insert has been generated, the other adapter sequence is actually complementary to the adapter 2 that is present here, right. So and then what happens is that because of the complementarity, this insert bends down and finds itself bound to the adapters 2 in this case. So another insert will start generating in the opposite direction. So this is an insert that is generated in a particular direction, say from 5' to 3' end of the fragmented DNA. Say that is a particular read. So we call this as a forward read. In the other case, when it is bending down to bind to the other complementary adapter sequence, right, another read is formed from the opposite direction, that is from 3' to 5'. So this is called as a reverse read. So in both cases, and this particular phenomenon is called as a bridge amplification. Basically you can see that it is kind of a forming kind of a bridge, right. So a bend bridge. So this is called as a bridge PCR amplification, and in this case a forward read in the same direction as of the fragmented DNA, and in the reverse read, that is the opposite to a direction of the fragmented DNA, is formed in this case. Basically is like a sense strand and antisense strand that is present within the DNA fragment there, right. So because it forms two different kinds of read, forward read as well as reverse read, it is called as a paired end read. So if only one type of read is formed, either forward or reverse read, then it is called as a single read there. But if two different kinds of reads are formed, it is called as a paired read. And if you look into pair read, there are both forward as well as a reverse read there. So I hope you get the difference there. And based on this, then another another extension is also formed there. The another reads are also formed there. Then these adapters can be removed from this in this case. And then another type of frequency sequencing can be done in this case. So this leads to a formation of what is called a cluster. So because several of the reads are formed within each flow cells, right. So it actually forms what is called as a cluster. So this is called as a cluster. So coming back into the introductory part of how these reads are formed, basically if you look closely, you need whenever a base is added, the sequencer actually processes it out. So what is happening is that the DNT piece, the nucleotide triphosphate that is added into a flow cell or the sequencing platform, is actually fluorescently labeled. So whenever a new base is added, here suppose that this is the first line, then you have to add the C. The C gives actually a blue kind of signal, right, light blue kind of signal, right, because it is fluorescently labeled. So whenever a C is added into this region at this particular foci, it gives a fluorescent signal, that is light blue kind of fluorescent signal. And afterwards adenine is present here, so thymine will be added complementary to it, so it gives a orange kind of signal. So and afterwards say cytosine is present here, so guanine is added here, so it gives a green signal there. And afterwards whenever a thymine is present here, adenine will be added and it will give a strong blue kind of signal. So based on the signal that is getting generated in an orderly manner, when the base is getting added into it, you can actually sequence it out. Basically what is added, like first it gives a light blue signal, meaning that it is adding a C, then a orange signal comes up, it adding a T, and then G is getting added up, so it gives a green signal, and A is adding a strong blue or deep blue signal is getting out. So based on this, what the sequencing does is that the sequencer actually consists of a high resolution camera that is present in it. So it clicks out each and every color that is popping up at different spaces in the flow cell. So they point out that okay, at this point this deep blue is getting out. So it maps up again at the same point what is the next color that is getting popped up, and afterwards what is the next color at that point is getting popped up. So it gets a photographic image of the whole flow cell, and based on the intensity that is getting or the color that is getting at different time points, it is mapping out the read. So that gives you an idea of which sequence is getting added and what will be the probable sequence of this particular read or the whole length read that is getting generated there. So from the color difference at different time point, you are generating the read sequence there. So basically, if you have fragment there, you can actually map out this particular read sequence or through the different intensity of colors that is generated in the flow cell and through the camera detector that is present within the sequencing platform. So this is how they are mapping out a read. Basically, this is how the process or the principle of Illumina sequencing is actually worked out. How a forward read and reverse read is generated there, what is a bridge amplification, and how the read is actually getting sequenced, and from the whole genomic fragments. So basically, this doesn't give you a whole genomic DNA sequence idea, but it definitely gives you an idea of the small fragments that have been generated from the as due to a part of a restriction reaction or a fragmentation of the whole genomic DNA. Then you have to mix all those read up, align it or assemble it to generate the complete genomic DNA. So you will get the read in the form of a file. So we have just downloaded that file outright. So we got the forward read as well as a reverse read from those both the SRA database using the SRA toolkit in the Bioconda environment and everything, right? So from that you get the reverse read and forward read. So you have to collect them separately, do a quality control. You need to exclude out some of the bases and everything, because what happens is that sometimes the signals tend to overlap with each other. Sometimes the signals will be slow to come up, or sometimes other signal sequence will be coming in front of her, mainly in case if in place of T, G will be coming up, or sometimes these kind of things do happen, or sometimes contamination will be there. And also as the porosity of polymerase decreases with the length of insert size, in this case the length of this particular insert size will be higher, then porosity will also get decreased, and these kind of things do pop up in this case. So we need to clear this out. So once you get the data of the forward and reverse read, that is the sequencing data from the SRA database or directly from the sequencing platform, any kind of sequence, whether it is .SRA, FastQ sequence, it doesn't matter. So then you have to do, once you have to got the data, sequencing data, you have to do a quality control, meaning that you have to remove any other genomic DNA contamination or DNA contamination other than the organism you are looking at. Then you have to do a primer removal, this adapter removal, because adapter is present here, right? We know the sequence of adapter. So if you know the sequence of adapter, then from the ends of the reads you can basically remove it out using computational tools and all. Then you need to do a quality control, because some of the reads that I will be getting here would have low quality, because most of the reads that has been analyzed, right, it gives a quality score. So the quality score determines how confident that particular read should be at that particular position itself. So you have to do a quality control based on that. You have to remove some reads, you have to trim some of the edges of the reads if at the ends it is causing any problem, and quality is less at the ends of the reads, forward or reverse reads. And then you have to assemble after doing a quality control, if you are confident enough that okay, this has been done properly, then you have to assemble the whole reads either to a reference genome that we know, or we have to do a de novo assembly, meaning that if you don't know a reference genome, then we have to assemble it by just doing an overlap match of the reads, and then from that generate the whole length of the sequence or the genome sequence, whatever it is, amino sequence that we are looking into. So there are two types of assembly. First is the reference genome assembly. If you know the reference, then you have to you can use the reference and then align the reads with each other to identify whether there is an SNP or whether this is the genome that we are looking at. Otherwise we have to do a de novo assembly, where if you don't have the reference genome, then we are doing a like overlapping kind of thing between the read ends, and then assembling the whole genome from itself. The quality control is good. So that we will all learn in this particular lecture video. So I hope this is understandable. So basically, like, if you have any doubt, you can just type in the comment box. I will clarify it. So yeah, thank you for watching the video.